So hopefully this is recording properly. What I'm going to go through today, we normally do this in a seminar. So I would have 2 to 3 colleges all together, and it would be more interactive. There'd be questions and answers. This is the best we can do this year, unfortunately. We've tried some seminars live, and you guys just don't seem to want to interact that much, and it's sort of understandable. Ideally, it's face-to-face. I'll try to not complain about that more during this, and I'll just go through the actual material. So this seminar is on receptors. Hopefully a lot of it will be review. And if it was live, I'd be able to ask questions. We'd know what level to pitch it at so that we could start where you are. We wouldn't bore people. We wouldn't terrify people. Because I don't have that feedback, what I'm going to do is just start with some very basics, build up, and by the end of it, you should be very aware of some receptor concepts. The ones that we're really going to focus in on are affinity and efficacy. If you understand affinity and efficacy, you'll understand all aspects of whether a drug is an agonist, a partial agonist, full agonist, inverse agonist, antagonist, any of these types of things. At the end, I'm going to add another layer onto this, whereby all the agonists and antagonists, the traditional ones, all act on what's called an orthosteric site. The orthosteric site on a receptor, if this is my hand's a receptor, orthosteric site is where ligands bind. The small molecule is called a ligand. This is a receptor. It's usually a protein. It will come in and bind to that site, but there's other sites. This is the orthosteric site. Other binding sites are called allosteric sites. This is going to be the future of a lot of drugs. Up till this point, most drugs are acting at the orthosteric site. So they're either hard on or hard off. Think of it as a light switch. Some of these allosteric site modulators, they're positive allosteric modulators or negative allosteric modulators. What they're going to be able to do is be like a rheostat, like a light dimmer, so that we can increase or decrease the output of a given receptor without using a sledgehammer approach of a pure agonist or antagonist at the orthosteric site. Don't worry, all of this will make sense by the end of this particular seminar. So the way I usually do this is I have 3 boards, whiteboards in pharmacology, and we go through each board. And I'll just give you an overview of what I would give on each board. I'm trying it with an iPad here. We'll see how this goes. It should show up on the screen. God help me, this records. I'll have to do it all again. So what I'm going to cover here, I'll say overview. So I can't, nope, I'm not sharing a screen. Okay, I'll turn on screen sharing. It's lucky I noticed that. Success. So this says overview. So the overview, what I'm going to give you information on in this seminar, it's going to be obviously about receptors, but I'm going to approach this in a different way than you're probably used to. If you're interested in more traditional ways, look at the textbook Pharmacology by Rang and Dale, etc., etc. It used to be just Rang and Dale. Now more and more authors are getting onto it. Maureen Dale, this person, I think is still a visiting member of the Department of Pharmacology. She used to be in the Department of Pharmacology, so that's one of the main reasons we use this. Rang actually did a lot of binding, radioactive binding back in the day, when people used to think that a receptor was nothing but a theoretical concept. He's one of the first people to do binding, and he used something called atropine from Atropa belladonna. Belladonna is a beautiful woman, and they used to use this in ancient cultures in Egyptian times. They would put extracts from this plant, Atropa belladonna. They'd swallow it, put it around their eyes. They would dilate the pupils because it's an antagonist of muscarinic receptors. So that's thus big dilated pupils make somebody prettier. Watch all the Disney or any cartoon animation things. Sympathetic characters, they always make really big dilated pupils, whereas unsympathetic ones, it's tiny little beady-eyed pupils. And people are very responsive to this. Even if you're not consciously aware of it, you'll respond to that. So he used atropine, Humphrey Rang used atropine, and it was radioactive. And he found binding sites. We'll talk about binding, we'll talk about affinity, we'll talk about what those curves look like a little bit. We won't go into super amounts of detail on that. So if you like a traditional approach, read, I think, like chapters 1 to 3. First chapters in there, it's been a little while since I looked at it. Anyone will do. Some of these principles haven't really changed in a very long time. So it doesn't matter which edition of this you have, look that up. The way I'm going to do this now is a little more non-traditional. I'm going to cover 3 areas at different levels, different concepts. The 3 areas are as follows. One's going to be at the level of signal transduction. Easy way of thinking of this is cartoons. So I'm going to draw what a G protein coupled receptor looks like, a G protein, et cetera, et cetera. And we may even, well, I will draw a G protein with 7 transmembrane helices and show you what actually happens when agonists bind, that sort of thing. So that's going to be the cartoon aspect of this. Second bit of this is going to be what I guess I can call mass action. So if you remember from chemistry, this is like reactants going to products. And if it's reversible, we put 2 arrows, and we can use this shorthand for talking about all the cartoon stuff. So it's shorthand, and we can put numbers to it. We can be quantitative like that. We'll be semi-quantitative in our approach today with this. We won't be fully quantitative. We're going to be just semi-quantitative with this material. Okay, in terms of the third one, it's going to be experimental. I'm going to give you sort of a whiteboard version of an agonist experiment. We'll throw in some antagonists. We'll throw in competitive and non-competitive antagonists. We'll look at what makes a full agonist, what makes a partial agonist. And all of these 3 concepts should fit together. You should be able to jump from one to another to the other and back and forth. They should all have meaning, and you should be able to interrelate them. If this doesn't work for you, go back to this textbook in pharmacology with Rang and Dale. They use a more traditional approach. So with that being an overview, actually there's one more thing I want to cover. Let's just think about what receptors are. So if we use Rang and Dale's definitions, there's 4 classes of receptors. We might as well start doing our cartoon type thing. So one class will be G protein coupled receptor, or GPCR. Get used to that. This will have an N terminus out here, a C terminus inside. It makes sense later when we go through how the structural changes occur when ligands bind to this. So that's the first one. Second one, let's see, I'm just going to I've erased more than I intended to do, but so if we've got this, second class is going to be a ligand-gated ion channel. and a small molecule, a ligand, capital L, will bind, it will open. Depending on the properties of the channel and the ionic gradients, it will let things in, things out. It might be cation selective, anion selective. These would be things like glutamate will bind to ion channels in the brain. So glutamate synapses, when you've got one nerve and another nerve, one releases glutamate, it binds to a ligand-gated ion channel, either the NMDA receptor or the AMPA receptors. If you don't know about those, you'll learn about them at some point. These are non-selective cation channels, and cations will flow into the cell, and you'll trigger an action potential. You'll trigger a depolarization. This could also be a chloride channel. There's a neurotransmitter called GABA, gamma-aminobutyric acid. GABA will bind to these ion channels, open up, and chloride will actually go into the cell. You should be able to think from first principles. If negative ions go into the cell, is that going to depolarize or hyperpolarize? And that's why these are inhibitory neurotransmitters, whereas glutamate's an excitatory neurotransmitter. So that's our second class, is ligand-gated ion channels. You should write the numbers down here. Third class then we can talk about is these go by various names. I think Rangandael says enzyme linked receptors. Most of these, painting with a bit of a wide brush, are receptor tyrosine kinases. So receptor tyrosine kinases are here. There's also some that have a guanylyl cyclase activity on the inner bit. So this is outside the cell. If I didn't say that earlier, this is out, this is in. And these will have some domain down here, some enzymatic activity. If it's a kinase, it will be a kinase. Kinases transfer the terminal phosphate off ATP onto a protein. Because this is a tyrosine kinase, it transfers it to tyrosine. My pen has now just quit working. Okay, I'm back. Battery problems. Damn pen. Its battery went dead. As Robert Burns said, the best laid plans of mice and men often go awry. When I do this live with whiteboard markers, usually not a problem, but now I'm having to deal with different levels of technology. So anyway, where I was talking about enzymatic activity, and there will be an enzymatic activity, I said here, could be a kinase on this receptor. That, because this is a tyrosine kinase, there will be the side chain on one of these amino acid, an amino acid side chain will be tyrosine, which has a phenyl ring, a benzene ring, and an OH off it. And that gets phosphorylated. Wherever you have an OH, you can phosphorylate. So other kinases do serine or threonine. This one happens to do tyrosine, and they're very important And that's an important criteria. You might think it's trivial, but the tyrosine kinase ones are the ones that are involved in growth and differentiation, meaning when they go awry, you get cancer. So there are a lot of problems with tyrosine kinases permanently being turned on. The cell runs amok. Big pharma, biotechs have put a lot of effort into tyrosine kinase inhibitors that end in nib. So like imatinib, the trade name's Gleevec. It's a tyrosine kinase inhibitor, a small molecule. Also, there's antibody therapies. Like antibodies have the two chains, so this is an antibody signal. It can go down and bind to this. These are functional antibodies. You guys will be seeing a lot more of those functional antibodies that turn off tyrosine kinase in cancer. Anyway, this gets phosphorylated, the tyrosine kinase. This serves as an adapter, and other proteins that have affinity for phosphorylated tyrosine will start snapping on the inner leaflet of the membrane. You'll get big signaling complexes that then go kinase, 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 kinase. You get all these cascades. Believe it or not, they're called MAP kinase, MAP kinase, kinase, MAP kinase, kinase, kinase. Eventually, it hits a transcription factor in the nucleus and drives gene expression, which gets me to the last receptor here. If we've got a nucleus, we'll have DNA in the nucleus, and we'll get transcription to RNA. This is dictated by, this is a transcription factor. And these transcription factors will bind a small molecule. Let's say it binds a steroid. So a steroid could start out here, come through the membrane because it's very hydrophobic. Hopefully you remember that from my lectures on drug metabolism. Goes down, binds to its receptor. That receptor then binds to the DNA and drives, well, it attracts RNA polymerase, and you will drive transcription. Hopefully you know all sorts of details. For all these different classes of receptors, you should know a handful of agonists, a handful of antagonists. You should know which receptors do what. So something like ligand-gated ion channels, an interesting one there is an antagonist called ketamine. This is a general anesthetic. It's a dissociative general anesthetic, meaning the person doesn't go unconscious fully, but they dissociate from reality. It doesn't cause breathing suppression. I'll come to that later in the context of some of the opiates. And it's also being used as a treatment for treatment-resistant depression. So if somebody tries a lot of different antidepressants and nothing's working, we call it treatment resistant, and ketamine seems to be like a miracle cure for this. They have to infuse it intravenously. Ketamine is also a street drug, special K. People go down the so-called K hole. They lose track of what's around them, so it's a non-dissociated anesthetic. There's other ones. There's benzodiazepines. My spelling's terrible, so look up the spelling. Benzodiazepines act on that GABA receptor, GABA-A, which is a ligand-gated chloride channel. And these are relaxants, but they're very addictive. I don't have time to go into why they're more addictive, but those would act on these ligand-gated ion channels. The G-protein coupled receptors, we'll cover that in a lot of detail in just a little bit. Tyrosine kinases, these are things like growth factors and cytokines, act on those. And nuclear receptors, these transcription factors, things like steroid-derived hormones. So cholesterol gets converted to steroid hormones, and then you have a lot of the female sex hormones, the male sex hormones, you have glucocorticoids, mineralocorticoids, all of these types of things. And think about the time frame of all these different receptors and where they are in the body and why they might be there. Things like ion channels, ligand-gated ion channels, they can open and close on a sub-millisecond time scale, meaning that you could fire perhaps 1,000 action potentials per second. It's really around about 200, but you need something really fast, whereas G protein coupled receptors, their times for action are seconds to tens of seconds to maybe 30 seconds. Think about an adrenaline rush. If you get scared, adrenaline's released, noradrenaline's released, it binds to G protein coupled receptors of the beta adrenergic set, and that elicits this fight or flight response. So think about how fast your heart rate goes up. A couple seconds, and then it takes a long time to go down. You can't use that as a neurotransmitter, but there are G protein coupled receptors that are used as neuromodulators. So they're called metabotropic G receptors in the brain, whereas the ones that deal with ion channels are ionotropic receptors. They both have different roles. One's very fast, one's quite a bit slower, but modulatory. So think about all the different times. Nuclear receptors. It took all of us a few years to go through puberty. These changes take a long time to manifest because you're dealing with growth, differentiation, gene expression. Tyrosine kinase is also, they are on the orders of seconds to minutes, but because they signal to the nucleus, they also are on the time scale of hours, days, months, those types of things. So think about the role of each one of these receptors, which tissues they're in. I don't have time to go into a lot of that now, but it would be quite a good exercise for you to look at G protein coupled receptors, and let's say adrenergic. And adrenergic we can then split, and hopefully this is very much known to you. There's an alpha and a beta subset of these. And then they found out that alpha splits into alpha 1 and alpha 2. And beta splits into beta 1, beta 2, beta 3. All of these have different selectivity for small molecules. So pharmacologists like these. We've exploited them for various medical treatments. So think about what tissues these are all in. So beta 2's in like lung and smooth muscle. Beta 1's in heart. If you ever forget which beta number goes with lung or heart, think how many hearts you have, think how many lungs you have. Therein lies your answer. Beta 3 is on adipose tissue or fat. Alpha 1's, they can be in a lot of places, but these can be on smooth muscle. And so can alpha 2, smooth muscle. Alpha 1's contract. Alpha 2's, I'll leave that up to you to think about whether they contract or not. A lot of neurons also have these types of things. Vascular beds, think about skeletal muscles, perfusion during fight or flight. Do you want to shut these down or turn them on, the vasculature? And then think about think about what agonists you know about these things. So let's say salbutamol. Think about antagonists. So for an antagonist, we have beta blockers that affect the heart, and there's whole families of these. The classical one is propranolol. Again, check the spelling on this. It actually blocks all of the beta receptors. The reason I mentioned that one is it was the first blockbuster drug. It made over £1 billion or dollars a year in sales. Sir James Black, a Scottish guy, got a Nobel Prize for developing propranolol and also antagonists to the histamine H2 receptor. So this is important stuff. And he got a Nobel Prize because he said you can separate affinity and efficacy, some of the stuff we're going to talk to you about in a minute. So play this particular game with this type of flow chart for also cholinergic receptors. Should be able to play that game for both of these at the level of detail that's appropriate for whatever you're studying. In terms of, now I'm going to try to go through my 3 different things. So I'll draw a line here and say, this is signal, transduction, or cartoons. And I don't know how much space I'm going to need, so I'm going to give it maybe one. We'll leave 2 just in case. And then here, I'm going to do my second thing, which is going to be mass action equations. I'll also leave, if I was doing this with boards, it would be much easier. I'll leave 2 of my screens for that. And then the third thing will be the experimental. So those are the 3 areas I'm going to cover, and I'll jump back and forth between these. So let's start with the signal transduction cartoon. So whenever I draw 2 lines like this, and most people, if you're a biologist, if you draw 2 lines like that, it indicates the plasma membrane. So this is the plasma membrane. And with the plasma membrane, then we can have an outside and an inside. We go over here and we'll draw our G protein coupled receptor. An N terminus, a C terminus. For an agonist, we just have to pick one and then stick to it. And I usually use acetylcholine as an example, and it will contract smooth muscle. So let's think about that. Next year, when you do some pharmacology practicals, you'll be doing experiments with this. I'll come to it more in the experimental system, but you'll use acetylcholine to contract. You'll use various acetylcholine antagonists, maybe even atropine, this from Atropa belladonna, and see that it relaxes smooth muscle. And you'll be able to determine whether it's a non-competitive antagonist or a competitive antagonist. But for now, let's just say we'll do acetylcholine. Acetylcholine is abbreviated A, capital CH. I'm not super big into abbreviations, but for these types of diagrams, it makes it a lot neater if we use a few abbreviations. So acetylcholine binds to the receptor, and then we'll have our receptor here, N-terminus, C-terminus, and let's make acetylcholine in a circle. So we've got a circle here with acetylcholine. It actually does bind relatively in the middle of all these helices going through the membranes. If you were a biochemist, you would call these 7 transmembrane receptors because there's 7 helices that go through the membrane. So now we've got it bound. So it's a G protein coupled receptor. So it's going to interact with the G protein. As an aside, these are called G protein coupled receptors because they bind guanine nucleotides. They will bind either GTP when they're activated or GDP when they're inactivated. I might say a few words about that, but not much. For right now, we're just going to say it's one lump. It's actually, they're heterotrimeric proteins. There's 3 subunits. Let's look at this first reaction, though, where we go from a receptor to agonist bound. Because pharmacologists are quite simple beasts, we call the agonist, the receptor agonist A, and we call the receptor R. So this would be a muscarinic receptor. Cholinergic receptors, the first division, goes to muscarinic, which are G protein coupled receptors, and nicotinic from the actions of nicotine. Nicotinic receptors are ligand-gated ion channels, whereas muscarinic receptors, depending on the cell type, depending on the tissue they're in, they can be metabotropic receptors in the brain, or they can be on smooth muscle. Depending on the smooth muscle, it might contract it. If this is gastrointestinal smooth muscle, acetylcholine will contract that. And we'll look at maybe the G proteins in a little bit. Right now, we're just going to say A for agonist and R for receptor, and that can take us then into our mass action equation. So instead of having to draw all these cartoons, this is shorthand for this. So we've got R receptor plus A agonist, and this will bind. So we have one arrow, and we go RA. As we showed on our cartoon, receptor binds agonist. Unless there's some very specific ligands, usually there's an off reaction. Sometimes it's very slow, but there's an off reaction. So we have 2 arrows here. It's in equilibrium. where that equilibrium lies, whether it's way pushed to the right for the receptor agonist interaction, that would be a high affinity ligand, or agonist. If it's pushed to the left, it's a very low affinity agonist or ligand. So what we're going to talk about is a constant called capital Ka, and this is also called affinity. And when we get to some of the later material when we're drawing the responses, we'll think about what affinity actually means. So this is how we would get to the point of figuring out the strength of binding. So now if we go back to our diagram here, what I can show is that just binding doesn't change the conformation of the receptor necessarily. So there's two steps. There's a binding step, and then does the conformation of the receptor change? The conformation of this receptor does change, and it actually looks quite a bit like this. We know all the X-ray crystallographic data from this. Lefkowitz and Kolbelka got a Nobel Prize in 2008, I believe, for the X-ray crystallographic structure. They know every atom in the beta-2 adrenergic receptor. So we now know what the inactive receptor looks like and the active, and it does kind of look like this. So normally when I'm lecturing, I can show this sort of more from an anatomical point of view. So if you imagine me as a G protein coupled receptor, if my left arm is the C terminus inside, if it's in like this up against the receptor, I cannot interact with a G protein, or imagine a rugby ball, can't catch it. If my arm's out here, if the C terminus is spun out, I can then catch a rugby ball. In our example here, the rugby ball is actually the G protein coupled receptor. So you can imagine that our G protein can interact with the open conformation of this, but it cannot interact with either the closed conformation, whether it's got ligand bound or not. So once it interacts with the ligand bound one, we have G protein that's bound to GTP. And this one would be GDP. So GTP goes on, GDP comes off, and that's a catalytic reaction that's catalyzed by the receptor. You can almost think of these activated receptors as having enzymatic activity. Every G protein that runs into them, they're going to switch GDP, the diphosphate, for GTP, the triphosphate, and then we get signaling. There are only there's three G proteins that you need to know about and two pathways. Memorize them. Just memorize them. It'll pay off dividends later. So in terms of our signaling pathways, we have GS and GI. The S stands for stimulatory, the I stands for inhibitory, and both of these interact with an enzyme called adenylyl cyclase. And this enzyme, it's actually an integral membrane protein, but we won't draw it that way. It takes ATP, makes cyclic AMP, and this then acts through a lot of different, what we call them, are targets. It can act through a lot of targets. PKA, protein kinase A. This is a kinase that phosphorylates threonine and serine residues, and it's a workhorse. All these pathways are common up to this point. Once you hit PKA, then all bets are off. It depends on the cell type. If this is in smooth muscle and you get PKA activation, you end up relaxing the muscle. If this is in cardiac tissue and you get PKA activation, it phosphorylates phospholamban, which disinhibits the calcium pump on the endoplasmic reticulum. It also phosphorylates a channel in the plasma membrane called the L-type calcium channel, which is also known as the dihydropyridine receptor. You get more calcium influx, you get more pumping into the ER, you get more forceful, faster contractions. So it's got positive inotropy and positive chronotropy. So GS stimulates this, GI inhibits it. So there's about 1,000 G protein coupled receptors in us humans. Compare that to about 20,000 genes in us humans. we're looking at about 5% of the human genome encodes for G protein coupled receptors. That's why we're going to be picking on these. I missed saying that earlier. We're going to everything I talk about now is going to be on G protein coupled receptors. Even though up here we said, oh, there's 4 classes of receptors, Rangendale says there's 4 classes. There can be. Everything that I teach you about G protein coupled receptors, you should be able to apply it to these other receptors. The reason we're focused in on G protein coupled receptors is that we know the most about them. We know the most about them because they're really biologically important, and they're also very important medically. About 25% of all drugs act directly on G protein coupled receptors. If you look at indirect interactions, it's upwards of 30–40%. Drug companies' profits come from about 60% of their profits come from G protein coupled receptors. So if you ever read reviews, they will say that 60% of drugs act on G protein coupled receptors. They're confusing where their profits come from versus how many drugs actually act here. They're also, though, super, super important. You go down to Boots, the stuff that you can buy over the counter, it's mostly hitting G protein coupled receptors. Some of the things I told you earlier, you should know agonists and antagonists. There's antagonists for the 5-HT receptor, sumatriptan, all these triptan type compounds, they antagonize 5-HT receptors, and they're very useful for combating migraine headaches. If you agonize one of those receptors, you can't buy this over the counter, but you can buy it probably if you know the right people on a street corner, LSD, lysergic acid, diethylamide acid, street name. It agonizes the 5-HT2A receptor and you get psychedelic experiences. So these G protein coupled receptors are really important. A lot of drugs act on them, so they're important medically. They're also important just from a basic biological point of view. If you follow this pathway from the point here of a receptor down through to protein kinase A, so all those different steps that you just glance at in a biochem or a pharmacology book and think, yeah, whatever. Every one of those steps pretty much got a Nobel Prize. So there's 6 Nobel Prizes from the receptor all the way down to protein kinase A. So these concepts are incredibly important just from a basic biological point of view. Getting back to our story about receptors, where we were going with these cartoons, we've now got, we've now got an activated receptor. And we call this activated receptor R, because it's still receptor, but then we say R star. So instead of having to draw all these transmembrane things, we can now just use R star. So if we jump back to our mass action diagram, we've now got ligand bound, but that receptor hasn't isomerized, hasn't done a conformational change. Then we can indicate its conformational change by going R star A. This is the active receptor. Then we can do a bunch of arrows. These are all the signal transduction steps down through pKa or whatever we deal with. And then we get a response. And this response will depend on the cell type, but we can measure that. So now let's go to an experiment where we can measure that. So if we're using, whoops, I just realized I forgot something back here. These 2 G proteins you do need to know about. The third one is GQ. GQ. This will also sometimes be called GQ11. And this protein interacts with an enzyme, not adenylyl cyclase, but something called phospholipase C. And that takes the phospholipid PIP2 and it will convert it, it hydrolyzes it, and we get IP3, which will then release calcium from the endoplasmic reticulum. ER here stands for endoplasmic reticulum. Diacylglycerol, the other part of this PIP2 that stays in the membrane, will activate protein kinase C. The C here, think of this being related to the calcium. Think of protein kinase A, this other one, the A relates to the A in adenylyl ATP AMP. That A stands for adenine, which is the purine nucleotide base on ATP. So pKa and pKc, that will get you really far if you just know those two. And remember, A is for cyclic AMP, C is for this calcium signaling pathway. So we're looking at GQ now, since we're doing, we're going to do acetylcholine contracting smooth muscle. So we're looking at GQ, and we end up getting a calcium release. For your purposes this year, anytime that you get a calcium increase, think you've got muscle contraction, be it in smooth muscle or striated. Striated is skeletal or cardiac muscle. Anytime you get an increase in calcium, we'll get contraction. So we went through some of our mass action equations. Now let's go to the experimental. And what we'll look at is a couple things, but let's say that we have force here, and this is time. So we'll have a certain force, then we can apply acetylcholine for the time of this little open bar. What's going to happen to force? If we were doing this interactive, we talk about this. What's going to happen is it's going to go up and stay up, and then when we wash it out, it will come down. So how do we actually do this? These will be some of the experiments you do next year. I'll run you guys through one. The one I demonstrate uses the hormone, the neuropeptide hormone oxytocin, which is released from the posterior pituitary, floods the system during labor in women, binds to receptors on the uterus and contracts the uterus. So we take the uterus out of a rat, string it up in an organ bath, get you guys to put oxytocin on it, see how to contract it, see how it relaxes by adding adrenaline, working through beta-2 adrenergic receptors, the same pathway that relaxes smooth muscle in the lungs. So I have asthma, so I took an inhaler just before I started giving this video. So hopefully I can breathe my way through the whole thing. It acts on beta-2 receptors. All that's for next year, but how do we actually do this? So I think it's maybe useful just to see a little bit of this. So if we have a tube here, so this is glass, so it's see-through, and it will have a port that goes out, and it will have a port that goes in, and we can then fill this with saline solution. So hopefully from your syllabus, you've memorized all those physiological parameters. You would know what saline should be in terms of sodium concentration, chloride concentration, potassium concentration, bicarbonate, all of that. So we'll have saline. Then we take a strip of muscle and it'll be in here. So this will be smooth muscle. And this will be from the ileum. So the small intestine. You'll be doing some of those experiments in your pharmacology practicals next year. Then how do we take thread and tie it off and secure it at the bottom so this doesn't move. Secured. We take thread, surgical thread, tie it off, put it up to the top, and put it on a force transducer. So this thing just measures force. Does the muscle pull or not? We hook that up to a computer, and the computer spits out this trace that I'm showing you here. That's what you would see on your computer screen. You will see force on the y-axis and time on the x-axis. So what we can do now is, let's say that we use 0.1 micromolar acetylcholine. We can go over here and we can jump up to 1 micromolar. We can jump up to 10 micromolar. We can jump up to 100 micromolar and 1,000 and 10,000 until acetylcholine stops going into solution. And what you will see with this is each time you go up, you get a higher and higher response, but at some point, it saturates. This is an incredibly important principle that separates basic chemistry from biology. Biology saturates, chemistry doesn't. So just as an aside, if you have reaction rate versus concentration, If this keeps going up linearly and it never stops going up linearly, you're dealing with a purely physical chemical process. If it goes up and then saturates, that's biology, because you've got a limited number of binding sites. This is pure chemistry. Of course, there could be more interesting things. It's out of fashion now. Growing up as a teenager, we used to make things that did this sort of reaction. As it goes up, it starts increasing exponentially. These are explosives, so you mix various things and you try not to kill yourself or kill your cousin, this sort of thing. It's good fun. Like I say, it's kind of out of fashion now. You probably end up going to jail, but we used to play around with those sorts of things. Anyway, back to fantastic. My eraser actually works. So back to the pharmacology of this. So we get saturation. Saturation is a biological type of thing. So we normally don't just use this kind of time-based curve. What we do is we plot the data, and you guys will plot some of these data. So I'll just make another plot here. And what we're going to do is do response. The response is going to go from 100%, 50%, 0. So we're plotting the percent response. This is a physiological response. That's going to become really important when we talk about efficacy. So with a physiological response, we could plot this in this is frustrating. Okay, I think I'm back. Having too many technical glitches today. So anyway, where we were was we were talking about we've got from 0 to 100. What you do there is you normalize this data. Normalizing means we take this and make it 0, and we take the maximum response equal to 100%. So the maximum response would be up here. Remember, it's saturating, so 10 is giving us pretty much the same as 100. And we could plot this linearly. So this could be linear on this axis, but we'd have to go out a very, very long distance. It doesn't make sense. So rather than linear, what people do is they do the log of this. Believe it or not, somebody's famous in pharmacology for logging one of these axes, because just one axis is logged and not the other. We call it semi-log. So this is a semi-log plot. Just the x-axis is logged, and we do log acetylcholine concentration. And this is where you can use 10 to the powers and do negative logs and all those sorts of things. Because I started using micromolar up here on our first plot, let's just stick with micromolar. So the plot's going to be logged, but the numbers are not logged, so technically it's not really log on here. It's just acetylcholine concentration, but I'm just going to leave that. So from this plot, this will be our maximum. This is our minimum. And pharmacologists are obsessed with halfway points. The halfway point here is not KD, the binding. It's something called EC50 for effective concentration 50%. So because this is a physiological response, the halfway point is EC50. All of these semi-log plots will look sigmoidal. It doesn't necessarily relate easily to affinity. That's one really important thing. Efficacy, this term efficacy relates affinity to the actual response. We'll go through that in more detail, but just make sure you keep those things clear and straight in your heads. So hopefully you've seen these types of curves, these semi-log curves. Note the saturation, note the halfway point. The other thing that we can take from these curves, you should have a rule of thumb with this. So if our halfway point, let's for the sake of argument say that it was 1 micromolar. What if we go 10-fold higher than that? All these curves work the same. So if you know the halfway point, if you go 10-fold higher, where does that get you on the response curve? So if we go 10-fold higher, so that this is 10 micromolar at this point, gives us roughly 90% response. I'll show you an equation in a little bit that you can get an exact value. It's either 89 or 91, I can never remember. But as a rule of thumb, if you go 10-fold higher than EC50, you should be at a response of 90%. The curve's symmetrical. We've got a symmetrical sigmoidal curve. So if we go 10-fold lower than the IC50, in this case it will be 0.1 micromolar, we'll be at roughly 10%. Again, it's 9.1 or 8.9, but it's around 10. So from this, we know if our agonist is behaving like it's got a single binding site. This is characteristic for a single binding site. If you don't get this, it doesn't mean that you're wrong. It means check your data. It can also mean something called cooperativity. If you go back to your notes, what you learned about hemoglobin versus myoglobin, look at some of these. Those plots are sigmoidal, but they're not semi-logged. They're sigmoidal because hemoglobin is positively cooperative. If I have time, I'll touch on that at the end of this. The take home here is that we can do an experiment. We can do a contractile experiment where we squirt in acetylcholine into this organ bath. This glass tube I showed you is an organ bath. We squirt it in, we wait for the contraction of the muscle. Then in their inlet, we can flush it with saline and the response goes back down to 0. Then we can add another dose and see what we get. And we should get an increasing response with dose. The increase in response with dose relates to pharmacology. There's a whole field of what I think is correctly called pseudoscience homeopathy. Don't have time to take shots at that right now, but basically the underlying message is homeopathy, you'd expect an increase in response with a decrease in concentration as they think things get more potent with dilution. Conceptually, doesn't make a lot of sense. But there's a lot of things that don't make a lot of sense. The world is actually round, but unless we go out into space, it's hard to figure that one out. It looks pretty flat if you were sort of somebody in the Middle Ages. Also, with homeopathy, it doesn't make sense, doesn't mean it's wrong, but the experimental evidence also supports that it's not wrong, that it is wrong, that it just doesn't stand up. Anyway, back to this. This is pharmacology. So we get these sigmoidal dose response curves. But like I said, we can't tell affinity, this term here, from the response. What that dose response curve was relating, we look to our diagram here, we were going from agonist concentration over to response. So it's purely dose response curve. And the halfway point on that was the EC50. But the EC50 doesn't necessarily relate to this affinity, this Ka, because there's a term efficacy. Efficacy describes, all the textbooks will tell you, don't think about it this way, it's incorrect for all these sorts of reasons. If you do graduate work in pharmacology, fair enough, we can have a high-level discussion about why it's not incorrect. It works for this. There is a guy called George Box, and his famous quip is, all models are wrong, but some are useful. This model is wrong, but it's really, really useful. We're going to say that we can separate affinity from efficacy perfectly, and we can put numbers to them. This is what got James Black a Nobel Prize. For the beta adrenergic receptor, he said, we knew that we could maintain binding, but we lost efficacy. That's how we made antagonists. They still bound, but they didn't have efficacy. Efficacy is how easily the receptor goes from this inactive state to the active state for activating a G protein coupled receptor. So that up here is this reaction, where it goes from the C terminus in to the C terminus out. That is the conformational change in the receptor that we relate to efficacy. And we can put a number on that. We usually put a coefficient. So this can vary from 1 to 0. So a 0 would be an antagonist, a 1 would be a full agonist. If we look at plots for this now, so let's go down to our experiment. Let's say that we did a plot, and I won't bother. The axes are the same. I'm not going to take time to draw all those out. Let's say that we got a response like this. Another molecule gave us a response like this. This will be a full agonist. This will be a partial agonist. Something that just hovers around the baseline would be an antagonist. And I now need to erase something. So we can think of efficacy as this coefficient between 0 and 1. Full agonist would be 1.0. Partial agonist, if I said I wasn't going to draw things on here, but let's do it. 100%, 50%, 0. So if this is 50%, we would say it has an efficacy of 0.5. We could multiply, if all the receptors were bound to agonist, they were saturated, but we only get half the response, our efficacy term is 50% or 0.5. For an antagonist, it's 0. We can saturate the receptor and we're never going to get a response. So think of efficacy as this coefficient, even though all the textbooks tell you not to, and it isn't perfectly correct, but our models are not perfectly correct with these. They are useful. Some of them are useful in George Box's terms. So what we're looking at here, this is a functional response. And this relates to something called EC50. But EC50 doesn't relate to affinity because there's this efficacy term that goes from 1 to 0. Efficacy relates to this response, the change in the receptor, usually. There's other people that have a different idea. There's Del Castella and Katz. I think this is their definition of efficacy. Stevenson was the first one to put this idea of efficacy out there. His is a little more simplistic, and it's not quite as good. This mass action diagram I'm showing you is Del Castella and Katz, that version of the world. If you look in Rang and Dale, I think they get into this in one of their sort of side boxes. The biggest thing is you cannot determine affinity from efficacy. Why would anybody want to know affinity? It used to be the only way that we pharmacologists could classify receptors was by affinity. But because agonists are complicated by this efficacy term, what they did was they used the affinity of antagonists. The way that you could directly determine the affinity of an antagonist is if you had it radioactive. So remember I told you Rang from Rang and Dale, this book, he used something called tritiated. This is H3, it's a radioactive beta decay form of hydrogen, and he used atropine, tritiated atropine, and bound it to muscarinic receptors. If you do that, you can actually get, then the equation is receptor plus atropine radioactive will go to receptor plus radioactive atropine. And you can measure this. So then you can get this type of curve where this is bound and this is concentration. and you can directly measure the affinity. People do this now. We do it in our lab. We synthesize little radioactive molecules and do radioactive binding. The paperwork's horrendous now compared to when Rang was doing this, unfortunately. From this, you'll get a halfway. You know it's halfway because you get so-called B max, the maximum bound. You'll get a halfway. Looks for all the world like our physiological response where we're looking at muscle contraction in an organ bath. But keep these things straight. What we're looking at here is antagonist binding. The curve looks the same. We get a halfway point. We get saturation because it's biology. But the halfway point here is called the capital Ka, or the dissociation equilibrium constant. So Ka is affinity. We can put a number on it by this equilibrium dissociation constant. This is with radioactive binding, and that's how receptors first went from this, as I said earlier, a theoretical concept that was just useful to actually being real physical entities, and they were able to work out some of these numbers. But before, with this, you'd have to make every single antagonist radioactive and do all these curves, and these days fill in all the damn paperwork. Back in the day, there was somebody called Churchill who came up with a different idea, and this will be Shelled analysis and competitive antagonists. So with competitive antagonists and Shelled analysis, you can calculate the affinity of an antagonist using just an organ bath experiment. This is phenomenal, and it's really cool, and like all the most cool things in the world, it's really simple and elegant. So what you can think of here is this is response. So this is not binding, this is response. It's a physiological response. So our halfway points are going to be EC50, the effective concentration, 50%. If we get this and we call it control. And then, I'm drawing this in the wrong section. Draw it down here. So this is competitive antagonist. Should be my experimental section, sorry. On the whiteboards, when we do this live, I don't have such problems. So this is response, physiological. Hopefully you're tired of me saying that. This will be log acetylcholine. So we get a response. This is our control response. Now let's chuck in atropine. And what you see, it's this plus, we get a shift in the response. We have to go to higher concentrations of acetylcholine to get the same response. So if this is our 50%, we've shifted the EC50 to a higher amount. Remember, the lower the number for EC50, the more potent something is. You need less of a concentration to hit the same response, in this case, 50%. So this is shifting the curve. What's really cool is we can quantify that shift in the curve, and that's what Shell did. So if I come back here, and I will erase some of this. So what I'll do is I'll redraw So we go receptor plus agonist gets into equilibrium with receptor agonist. And then if the agonist changes the receptor into the active form. Oh, forgot one analogy that some students find useful. I find it useful to think about this stuff too. Think about it as your college key. If you have a really good key, it fits into the lock. Fitting into the lock, think of that as, does it have affinity? If you take your college key and come to the Department of Pharmacology, probably won't fit into the lock. If you try other people's rooms in the college, they'll probably fit into those locks because the side profile will allow it to fit in. But it will only turn in your lock. If you try it 10 times and every time that you try to put it in and twist it, it's bound and it has efficacy. If it turns, it has efficacy. It turns on your lock just like the agonist is turning on the receptor. If every time you turn it, you get the lock to turn and the door opens, you've got a key with 100% efficacy. It's a full agonist key. If it only works 1 out of 10 times, it's a partial agonist with that efficacy coefficient of 0.1. You can have anything in between. If you accidentally stole your neighbor's key and try it and it never works, you've got efficacy of 0, you've got an antagonist. All of them bind, all of them fit into the lock, but only some of them turn the lock. That's like what these ligands are doing with these receptors. If they activate the receptor, go to R star, chuck out that C terminus, they activate it. If they don't, they're just blocking other things from acting. The blocking is due to competitive antagonism at this orthosteric site. So they're binding to the same site on the receptor molecule. So we said that we can't, on this equation that I've just drawn out again, we can't get Ka from this because of this damn efficacy term, which we don't necessarily know. We can estimate it, but it still doesn't help us calculate the real affinity. But from what I showed you on this other set of curves, we can add an antagonist and it shifts the whole dose response curve. Let's think about what we're doing. A competitive antagonist to a pharmacologist, we label it B for our mass action diagrams. So R plus B, going to an equilibrium, RB. But this, it doesn't, antagonists have efficacy of 0. So it's a dead end complex. So nothing happens. Other than we've got all these linked equilibria. If you remember a principle, Le Chatelier's principle, if you have linked equilibrium, if you push harder on one end, it will shift through all the equilibrium. If you pull on one end, it will shift everything back. So if you add something, it will shift it. We can predict what that is. So if we're adding a certain concentration of B, our antagonist, it's going to shift this whole equilibrium down to this RB complex. And the amount that it reduces the complex, the concentration of the R star A complex will relate to the amount of the response. Now, you guys won't have to know this. If you have to work through a problem set, this will be given to you. But there's something called R subtract 1 equals B over KB. And we can even put that in concentration. This is a Scheld equation. And with a little rearranging, this is the same equation for biochemistry. If you're looking at enzyme kinetics with Michaelis-Menten plots, this will give you a Ki, which is the inhibitory constant for enzymes. It's the same thing. Single binding sites are all the same thing. If we take logs, if we rearrange these equations and take logs of them, we can even derive the Henderson-Hasselbalch equation, because with that you've got a base and it's binding to a proton. There your receptors, the base, the protons, the A ligand agonist, and everything works out. Everything boils down to this equation for single binding sites. Anyway, this Scheld equation, we can relate R, which is the dose ratio, which means, back on this one, if the shift was 10-fold, let's say 11-fold, then we would have 11, R11, subtract 1, is equal to B over KB. So then we could chuck in some numbers. Let's say we would know this. If you did the experiment, you will have weighed out some powder, atropine, and if you got your calculations right, you'll know what concentration of B you put in. So that's here, atropine, certain concentration. R, we can get from plot, from our actual experimental plot. And 1 obviously is there. This allows us to calculate B. And B, all we're looking at is the reduction in the activated receptor. So KB on this particular plot is at this equilibrium. So we can calculate KB, an equilibrium dissociation constant, from a physiological experiment, pharmacological experiment in an organ bath. This revolutionized pharmacology. Again, you might be thinking, well, who the hell cares? Why do we want to know a KB? Receptors used to have to be characterized by their affinities to a lot of different antagonists. So we would be able to characterize the M1 muscarinic, M2 muscarinic, M3 muscarinic, M4 muscarinic receptor based on their KBs, their affinity for various antagonists. And that was all through this shelled analysis. So even though we can't directly know the affinity of an agonist, we can calculate an agonist dose response curve and use it with an antagonist to get the affinity of the antagonist, because antagonists don't have any efficacy. In terms of other types, well, we can get families of these dose response curves, where this is B1x, B concentration at 10x, B concentration at 100x. And each one of these shifts, we could plug into our Shield equation, and you can do Shield plots, and it gives us other information. Unless you start doing graduate work in pharmacology, don't worry too much about it. But this pattern of a rightward shift, so for a competitive antagonist, it should be right shifted. It's right shifted because we can play a game with these dose response, with the mass action. Imagine that we're increasing, this is getting a little messy now, but look here. If we increase A, through Le Chatelier's principle, we'll shift the equilibrium more to the R star A. So we can keep playing this game where we add one concentration of antagonist, do a complete dose response curve. At a higher concentration of antagonist, do a complete dose response curve, and so on until we get a family of these curves. The characteristic for a competitive antagonist is, as I say, it's right shifted. Also the maximum, it goes to the same max. We call this, it's surmountable. Same max response. And the shift is parallel, meaning for every point on this curve, it shifts the same. Every point on this curve, the shift is the same. So you get a parallel shift in all these. That's competitive. If we have non-competitive, this is our control. We would get a family of curves where we get decreases in the max response. These are non-competitive. You can also get competitive irreversible. So this is where your antagonist forms a covalent bond. So we'd have R plus, well, I'll do it the other way. Or actually, I can do it this way. I won't bother with all that stuff. You should be used to it. Let's be this. Notice this is one way. The arrow is just going one way. We form a covalent bond. This will permanently shift the number of receptors, thus decreasing the maximum response. So that's a non-competitive or a competitive irreversible. Non-competitive is usually binding at a different site. So if we have a GPCR, and this is the orthosteric site, that's where the competitive irreversible or the competitive antagonist would bind. So B could bind there, or A could bind there. We could have something called C that binds to another site here and just alters it. That's how we get non-competitive. They're allosteric sites. And like I say, that's the future. Keep an eye on that space. In terms of, let's see, hopefully you'll have a very good feeling for R plus A goes to RA, R star A response plus B, RB, no response. Or we could have R plus B going all to RB. know that this efficacy, this will explain all agonists and antagonists that bind to the same orthosteric site. We could get ones that bind to an allosteric site and it will modulate this. Relate this to responses like this: pure agonist, partial agonist, antagonist. There's also something here, an inverse agonist. If we've just got our idea of R plus A goes to R, I'll just put star A now, we get an instantaneous activation of the receptor. How could an inverse agonist work? So if we normalize all the data to 0, imagine this is the amount that smooth muscle has contracted. There'll be some tone. It will be not necessarily completely zero and flaccid, not necessarily completely relaxed, but we take that as our baseline measure and we call it 0. Then as we add some compounds, this is, oops, this is log compound concentration. As we add log compound, we get a decrease. This is an inverse agonist. Our idea of small molecules binding and then getting a receptor change, conformational change in the receptor, had to be modified at this point. The model, remember, all models are wrong, some are useful. The model that they now like for describing inverse agonists is R is actually in equilibrium with R star, the active receptor and inactive receptor. It's always flickering between these states. In most cells, the equilibrium of this is shifted mostly to the left in favor of R, but there'll be a little bit of R star. This gives us what we call constitutive activity. An inverse agonist binds to R star, IR, I'll abbreviate inverse agonist. This gives us, or whoops, sorry, wrong way. Inverse agonist binds to the inactive receptor and it forms an equilibrium and we get IAR. An inactive receptor bound to the inverse agonist. Through Le Chatelier's principle, it will shift the whole equilibrium from R star to R, even though R star didn't have any ligand bound. That explains inverse agonism. So for an agonist, we would have it binds to the R star form. It's got greater affinity for R star and it shifts the equilibrium towards that. Le Chatelier's principle means that we get R shifting to R star, we get a bigger response. What about the complicated one? Well, the one that strains credibility is the pure antagonist. With this one, we would have to have, we don't want to alter, you can think of this as a ratio. So a response would be how much R star over R. That will give us our response. An antagonist here is neutral. It's got efficacy of 0, so it can't alter the ratio of R to R star. So what we say is it has equal affinity, exactly equal affinity to both the activated R star and the inactivated one. As we investigate a lot of what we thought were pure antagonists, they turn out to be inverse agonists. There probably are by chance a few pure antagonists, but a lot of them are inverse agonists. This is called, because we have R and R star, it's called the two-state model. And at this point, so we've got either R and R star already exist, or we've got R binding to something and it induces a conformational change to R star A. And students always ask, which is correct? And the answer is both. Depends how you look at the question. All models are wrong, some are useful. If you guys are interested in it, go and look on the internet. A guy called Leonard Susskind, a professor at Stanford University, came up with string theory. He gives a lot of lectures he calls physics for old farts. go on YouTube, watch some of these. He talks about black holes. And in one version of reality, you get sucked into a black hole. If an outside observer, you die. In your realities, you're sucked into a black hole, you're alive and nothing has changed. Depends on the observer's point of view. In terms of pharmacology, it depends on what question we're asking. There is no right or wrong with this. You need the right tool for the job. You need the right model for the job. The reality, now that we're getting X-ray crystallographic structures and cryo-electron microscopy structures of G-protein coupled receptors, certain receptors show one behavior where they have to have this induced conformational change. Other ones show that conformation already exists and has greater affinity. This should be very familiar to any of you that did a higher level of biochemistry with two models for hemoglobin interconverting, whether all the subunits interconvert at once, the concerted model, or there's the induced model where one oxygen binds, tugs on the other subunits, causes them to increase their affinity. So there's no real right answer. Depends what you're looking at. One model works better sometimes. The other model works better sometimes. Just to end with, I'm just going to give one sort of example that will hopefully give you another hook to hang some of these ideas on. It's going to be more concrete than something that you'll do next year in an organ bath. If we have, again, two parallel lines means a plasma membrane. Here I'm just going to go G-protein coupled receptor as a rectangle. And this G-protein coupled receptor we're going to look at, it's going to be the mu opiate receptor. So here it is, it interacts with G-protein called GI. GI, I've already said, interacts with the genital cyclase. And this, in a nerve, the concentration, so this is a neuron. In a nerve, the concentration of cyclic AMP, you can think of it roughly like this. If it increases, then increase in firing, increase probability of an action potential. If decreases, then decreases rate of firing, decrease in the probability of an action potential. So GI inhibits genital cyclase, so cyclic AMP levels drop, pKa activation drops. We'll just say targets at this point. We can go through that in more detail next year at some point. So it decreases neural activity. Same thing's true in heart cells. Cardiac, ventricular cells or sinoatrial node cells. Cyclic AMP relates to excitability. So that's why it's kind of useful knowing this pathway. Then in any electrically excitable cell, be it cardiac or neurons, you can say cyclic AMP, if it increases, it will increase this. If it decreases, it will decrease this. For opiates, you have endorphins, endogenous opiates. These are peptides, they will bind. However, usually we use morphine. So a very good drug for pain control, probably the best pain control medication we have out there. It comes from the opiate poppy. Humans have used this for 10,000 years. It shows that you don't need to know the target of a drug to have a very effective drug. They'd make tea out of this and it worked really well. As Western medicine approached it, we isolated morphine, we acetylated it, made it greasier so it crosses the blood-brain barrier faster. And that greasy form of morphine is called heroin. And it's very addictive. The old teas probably weren't very addictive. The faster you can get psychoactive substances into the brain, past the central nervous, into the central nervous system, past the blood-brain barrier, the more addictive they will be. So one of the problems with this, one of the downsides of morphine and opiates in general, there's a lot of synthetic ones. In fact, I'll list one. Fentanyl is the one that's killing a lot of people nowadays because it's about 100 times the potency of heroin. So very small amounts, you can get the dose wrong. And the dose doesn't affect necessarily the pain killing. Let's look at that for a second. So if any of these opiates bind to the receptor goes to R star, it activates the G protein, decreases cyclic AMP, decreases neuron activity. If this is a pain neuron, this is what you're looking for. You want it to reduce pain. If this, however, unfortunately, is a breathing neuron, some of the neurons that go from the substantia oblongata, you have to memorize this, part of your brain stem that sends out motor signals through motor neurons to your diaphragm, controls breathing with your diaphragm. Those neurons also have mu-opiate receptors. So you're decreasing the activity in pain neurons, but you're also decreasing the activity in the breathing neurons. So people, when they die from an opiate overdose or they're overdosing, they get really shallow intermittent breathing because of the effects of the opiates on this. This is through a G protein coupled receptor. For luck, do not forget this name. This name of this drug, well, it's not on your list, not on your syllabus list, but this will save somebody's life. Naloxone. It's a competitive antagonist. So we could do R plus A goes to RA, and plus B goes to RB. We know that we can push-pull on this. So if you give a large dose of naloxone, it's going to outcompete all whatever opiate is there, and this person will, their breathing will come back. It also relieves other symptoms of this. They're probably opiate addicts. It probably makes them go cold turkey. That's so called because you get a bounce, an increase in cyclic AMP. It's like if you've ever drank too much coffee, this is like drinking 100 times that, and people get piloerection. So all the hairs on the person stand up. It's from this excited state. That's why it's called cold turkey, because you look like turkey skin at that point. It's very unpleasant. The stories might be apocryphal, I'm not sure, but some of my clinician friends who worked in A&E said that people would come in, they're an opiate overdose, they're not breathing, they inject naloxone, they come out of it almost immediately. It's a very fast response. They're feeling like crap because they're opiate addicts. They check themselves out of the hospital, they make it 2-3 streets away, they pass out again. The metabolism of morphine or heroin or fentanyl is very slow relative to the metabolic rate of naloxone. Those drug metabolism lectures that I was telling you about, this is a practical application of that. So what they do now is give naloxone and they titrate it in, and they also tied in benzodiazepines, what I mentioned at the beginning of this particular seminar, which I guess has turned into a lecture this year. Benzodiazepines act on the GABA A receptors, which are chloride channels. So you're giving somebody a sedative, which seems really weird, but you want to keep these people calm, even passed out indeed, so that the whatever opiate they've overdosed on can be metabolized. There's also a longer lasting form called naltrexone. And that doesn't seem to be used as much, even though it's longer lasting. People I've talked to said they can't titrate the dose quite as much, so they don't like using that one. Whereas naloxone's faster on, faster off. They can control the dose. There was something else I was going to say about this. Yeah, the fentanyl crisis is killing a lot of people. In the States, I think is between 60 and 100,000 people a year are dying from fentanyl. What happened was GPs, well, traces back to somebody put a 5-line letter to the editor in the Lancet, I believe it was, or the New England Journal of Medicine, saying that people don't get addicted to opiates if you're using them for pain control. Pharmaceutical companies picked up on that and they started giving slow release opiates. And they got a lot of people addicted. So just simple back pain, I say simple, it's not necessarily simple, but things that they never used to use opiates for, they were then using opiates for. People got addicted, government stepped in, and the GPs said, oh, you shouldn't be on these anymore, you're getting addicted. These people had to turn, because they've got an opiate addiction, which is very hard to get over. The drive is incredible from what I understand. These people turned to street dealers. The street dealers were getting fentanyl. Fentanyl's really potent. It's so potent, most of the fentanyl comes in from other countries, labs in China, India, and they just use conventional letters in the post. The US Post Office, its rules are, I think it's less than 185 grams, they're not allowed to open a letter. So even though they think it might contain fentanyl, they're not allowed to. The head of the post office in the States said that the US Post Office is the biggest drug distributor in the world now of illicit drugs because of this, because fentanyl is so potent. The other problem with fentanyl being potent is that 1 grain on the tip of a pin will take the edge off your addiction. 2 grains can cause respiratory depression and kill you. This is why so many people are dying from this. Naloxone in Canada, at least, people have decided to go for harm reduction. So you get an EpiPen-like thing, but it contains naloxone. So this is being given to police and emergency responders like ambulance workers and firemen. And it's all for harm reduction with this. I think it's a very good idea. There was one other thing I was going to tell you about here, and I can't remember it. Should be able to play that game. You should be able to understand why if one gets metabolized faster than the other, you get this push-pull. I think that is it then. So just in summary, what I've told you about is receptors. Oh, I know what I was going to tell you about. We normally think if these are GPCRs, let's say this is the 5-HT receptor. So this is the one that responds to serotonin, 5-HT. And let's say we have CB1, an interesting one. This is the cannabinoid receptor. This one normally has its effects through GQ, calcium, etc. This one's usually through GI. So you can imagine if you use delta-9 tetrahydrocannabinol, if you smoke marijuana, hashish, if you take forms that are more medicine, drops under the tongue, the THC is very hydrophobic, so you can get absorption from that. Because it's GI, neurons that have GI, they will decrease their activity. That's how it works. If, however, what we now know, these receptors can dimerize, and all bets are off. Suddenly, you might be signaling through GS, GI, GQ. You don't know. And so cannabinoids that normally signal through GI and decreasing cyclic AMP may then start signaling through 5-HT. If it's 5-HT, two receptors, 2A, this is the same effect as LSD acid. So you can get psychotropic effects. These receptors, like I say, there's about 1,000 GPCRs in us humans. A lot of them can dimerize with each other. It changes which second messenger pathways they interact with. This is so scary and depressing. People are kind of, pharmacologists are just putting it to the side. We don't know what to do with it. The other thing that relates to this is you can get receptor dimerization, and it can completely change ligand specificity. So if this receptor normally would respond to sweet, so our taste receptors are GPCRs, this would respond to sweet. This one would be umami. This is basically savory. MSG, monosodium glutamate, will hit this one. This one's type 2 and type 3 taste receptors. This one's type 1 and type 3. Notice the type 3 can participate in either sweet signaling or umami signaling, depending on how it's dimerized. We're starting to find this for other types of receptors. Also on the sweet receptor, there's, let's say, these are orthosteric sites. So this is where sucrose or whatever artificial sweetener you want bind. There's also allosteric sites. There's allosteric sites also on umami. So MSG got a really bad rap a few years ago, so nobody wants to put MSG in their food anymore. It's all kind of a myth. I don't have time to go into the details. Look it up if you're interested. What food manufacturers do is they put mononucleotides in. GMP, IMP, inosine monophosphate, and I think it's AMP. They're monophosphates, I can't remember for sure. These bind to other sites, they're allosteric modulators. So if this is normally your MSG response, and this is taste units, you get this physiological response. If you put in IMP, it actually shifts it this way. So these are positive allosteric modulators, and they shift the curve. So the natural amount of glutamate or monosodium glutamate, you then detect it. Companies look at crisp packets. They will say these are preservatives. They might be, I don't think they are. They're not really single electron scavengers or anything. They put them in to shift the dose response curve. They were way ahead of any of the pharmacologists with this. We pharmacologists are just getting into this. So look to the future for dimerization of G protein coupled receptors and allosteric sites. I didn't say anything about the other receptor families, but everything I've said about GPCRs apply to the other receptor families. So if you have a ligand-gated ion channel, something's got to bind, its conformational change is opening of the channel. If you have a steroid receptor, it will have to bind DNA. So whether, once that binds, whether it binds DNA or not, that's its activation, that's its R-star equivalent. So hopefully this has made sense. If it doesn't, this is very much building things up from a conceptual point of view. It's the way I view the world with this. If it doesn't make sense at all, my apologies. Go and read Rang and Dale, and they'll cover everything in a very conventional way. Hope everybody's doing well. Take care.